Chapter 27 is all about our favorite prokaryotes, the only prokaryotes, Bacteria and Archaea, and we're going to learn all about their structure, how they reproduce, all the ins and outs of these prokaryotes. We divide prokaryotes into two categories, the bacteria, which you would see living absolutely everywhere, and archaea, which can be found in extreme environments, such as places that it's too acidic, salty, cold, hot, too little oxygen for any other organisms. There's more prokaryotes in a handful of fertile soil than the number of people who've ever lived on the face of the earth. And they have a lot of diversity amongst them, despite being only single-celled. They're very different. There's a lot of genetic diversity within this domain. Let's start off with the structure of prokaryotes. They are mostly unicellular, although we do see some of these unicellular bacteria come together and they will live in a colony, so they look multicellular even though they're individual organisms. They're really, really small. We're talking 1/10 of the size of a eukaryotic cell, and they have a variety of shapes. Prokaryotes can be spheres, rods, or spirals. Here are images of what those prokaryotes look like. So they can be spherical, which is also called cocci. If they form chains, they are streptococci, which you are seeing here. In the image, they can be rod-shaped, which is also called bacilli, and they can be spiral-shaped, and you're looking at a spirilla in this image in the last on the right. Prokaryotes have cell walls, and the function of those are to maintain the shape of the prokaryote, to provide physical protection, and it also allows the cell to prevent any bursting if the cell is put into a hypotonic solution. You have water coming into the cell. Eukaryotic cells, if they're plants, they're made of cellulose. They can either be made of cellulose or chitin if we're talking about an insect eukaryotic cell. But bacterial cells are different. Their cell walls are made of peptidoglycan, which is made up of both carbohydrates and proteins. So you have these sugar polymers, that's where you get the glycan part of the word, that are cross-linked by polypeptides, which are your amino acids. Gram staining is a technique developed by Danish scientist Christian Gram, and what it does is it takes this crystal violet dye and it washes it over your bacteria, and if the crystal dye stays and it looks more purple, you have a gram positive sample, and if the crystal violet washes away, you have a gram negative bacteria. And that is one way that we classify our bacteria. They are either gram positive or gram negative. If it's gram positive, we're talking about much more simple walls, and they have a lot more peptidoglycan, which picks up that gram staining. Gram negative has less peptidoglycan, which is why they don't hold on to the gram stain. And they also have an outer membrane that can be toxic. These guys are the ones that are tougher to treat if you're infected with a gram negative strand of bacteria. They tend to be antibiotic resistant. A lot of antibiotics that we have on the market today target the peptidoglycan in the cell wall of the bacterial membrane. What they're targeting is breaking down that cell wall, which would in turn lyse the bacterial cell, killing it. Here's an image showing how gram staining works. So this is your peptidoglycan layer right here, and if it is a gram-positive bacteria, they are going to take in the crystal violet and the red saffron, which is another dye that is used in gram staining, and the crystal violet will not easily rinse away. Your red saffron is going to stay in there as well. In a gram negative bacteria, you have a much thinner peptidoglycan layer, and because of that, your crystal violet will rinse away pretty easily, leaving just the red saffron visible, which gives it kind of a reddish color. So here I'm seeing gram negative bacteria, and this darker purple we are seeing gram positive bacteria. All right, you're looking at Streptococcus bacteria. This is a prokaryote, and on the outer surface of that prokaryote you have what's called a capsule. And a capsule is a polysaccharide or protein layer whose function is to protect the cell from drying out. But in this particular species, the function of that capsule is to allow it to adhere to the surface of the respiratory tract. So you would find this particular bacteria infecting the respiratory tract of an organism. Some prokaryotes have fimbriae, which is also called attachment pili, and these guys function in the same way that the capsule does in other prokaryotes, and it allows for these prokaryotes to stick to their substrate. It also allows for other individuals to stick to one another and to form a colony. Sexpili are longer than Fimbriae and allow prokaryotes to exchange DNA. So you can have the exchange of DNA in two adult cells, creating genetic variation between prokaryotes. If your bacteria is motile, it can propel itself through an environment using flagella, which is structurally and functionally different from a eukaryotic flagella. It is an amazing motor. It moves super fast. A prokaryote can move at 50 micrometers per second, which is not really that fast to us, but the equivalent for a person my size would be running at 306 kilometers per hour. So that's pretty speedy if you consider the ratio. If there's a heterogeneous environment, bacteria can exhibit taxis, which is movement toward or away from a stimulus. Here's what your flagella would look like close up. You have a ATP-powered proton pump. So ATP is pumping hydrogen ions. A pump is pumping them out of the cell, and as they diffuse back in, that's what powers your basal apparatus, which is what allows movement of the flagella. Prokaryotes have unique structures for their genomes. They don't have a nuclear envelope that holds in all of their DNA and their genomic information. They don't have any compartmentalization in their cells. They do have specialized membranes that can perform metabolic functions, and their genome tends to be circular, and it has a lot less DNA than the eukaryotic genome. The DNA, as I said before, doesn't have a nuclear membrane, but it does have what we call a nucleoid region where you would find that DNA. And some species of bacteria also have a separate, smaller ring of DNA, and that's called a plasmid. And this is what it would look like in a bacterial cell. So these really long little strings are the bacterial cell's chromosomes. And then up here in the corner, I can see 1, 2, 3 of those smaller circular pieces of DNA that we mentioned. Those are the plasmids. And so you'd have both the chromosomes and the plasmids as part of your prokaryote's genomic information. Prokaryotes can reproduce really quickly, and they reproduce through binary fission, which creates an exact copy from the parent cells. Now you have two daughter cells that are exactly the same to each other, and this can happen so quickly. It happens every 1 to 3 hours. So generation times are really short. And they can evolve very rapidly because of the short generation times as well. So if you have random mutations, those tend to accumulate pretty quickly because the generation time is only about 1 hour to 3 hours if the conditions are right. To demonstrate the rapid evolution that's seen by prokaryotes, scientists developed an experiment where they allowed the same strand of E. coli to grow in 12 different test tubes over the course of 3,000 days. So we are talking about 20,000 generations over that time. And in each test tube, they gave them challenging environments. So there was very little glucose and other materials that they need. And what they saw was they were selecting for organisms that could live in an environment with little glucose. And they found that their fitness relative to the ancestor, the original strand, increased over time. So the population was evolving based on the environmental conditions that they laid out in the experiment. There are 3 factors that contribute to prokaryotes' genetic diversity, and they do have considerable genetic diversity. And the first is rapid reproduction, which we talked about in the previous experiment, as well as mutation and genetic recombination. So we know that prokaryotes reproduce using binary fission. They copy their DNA, they double in size, they split in 2, and your offspring is going to be identical to your parent cell because of binary fission. Mutation rates during binary fission tend to be low, but because these guys can reproduce so quickly, the mutations can really accumulate rapidly in a population, especially if they're considered beneficial mutations. High diversity from these mutations allow for rapid evolution of prokaryotes. There are 3 different ways that prokaryotes can become genetic recombinant, so different from their parents and different from each other. And the first is transformation. So this is where a prokaryote takes up DNA from its environment. Second is transduction. Your virus is transferring genes between prokaryotes from one prokaryote to another. And the final one is conjugation. This is where genes are directly transferred from one prokaryote to another. Here's an example of how transduction works with a bacteriophage, which is a virus that specifically infects bacteria. So here we see the virus is infecting the cell and it's inserting its viral DNA into the cell. That viral DNA is going to function in making new viruses, copying itself, and in the meantime, it also is going to chop up the host cell's DNA. So this cell originally had A positive and a B positive allele. When those viruses get packaged up, some of that original host cell's DNA can get packaged into the protein capsid of the virus. If that happens, like we see right here, we see the gene for A positive got packaged into the virus, and that virus then infects another cell, it's going to inject that original host cell's DNA, that allele for A positive, into the new cell that it just infected. When that happens, we see that this brown gene that was from the original host cell, that gets incorporated into the new cell's DNA, and that is considered a recombinant cell because now it also has the allele for A positive where it did not in the beginning. Prokaryotes can also go through conjugation to get genetic diversity, and in conjugation you have a sex pili that allows two cells to connect and they pull together, and then you can swap DNA doing that. A piece of DNA called the F factor has to be present in order for a sex pili to form. The F factor can exist as a separate plasmid or as DNA within the bacterial chromosome, and we're going to see a picture of what I'm talking about. All right, so here's an image of two prokaryotes, one of which is producing your sex pili, and it's connecting one prokaryote to another prokaryote. Once they're close enough, you're going to get a mating bridge that forms, and that's how the DNA transfer is going to happen. So this F factor is really important in conjugation because it determines who's going to be the DNA donor and who's going to be the DNA recipient. So if you have an F plasmid as a prokaryote, you are the DNA donor during conjugation. You are giving DNA. If you do not have the F factor, you are the DNA recipient during conjugation. The recipient is the one that becomes the recombinant bacteria because he is receiving new DNA that makes him different from his original form. He is now recombined. He has DNA from two different cells. The F factor can be transferred during conjugation as well. So an organism that was the DNA recipient without the F factor during conjugation can receive the F factor and then become a donor itself. All right, so we're going to see two different ways in which conjugation can occur, one in which your F plasmid is transferred to the recipient cell and one in which it is not. So in this top example, we see a donor cell who has the F plasmid, and that F plasmid is going to break at a certain point. So it's going to start to kind of unravel, and it will cross through that mating bridge that it has produced because it has the F factor in it. As it starts to leave, that causes your F plasmid to replicate itself so that it has a full set of the F plasmid again. It will cross through the mating bridge and into the new cell, and it reforms in a circular plasmid. So now both cells have the F plasmid, and both can be donors in the next conjugation event. The second thing that can happen is where you transfer part of your genes from donor to recipient, but you don't transfer the F plasmid. So here we see our F plasmid is actually part of our circular chromosome. This is an HFR cell, so F plasmid is part of that chromosome. A mating bridge is going to form between two of these prokaryotes, and you're going to get a break in your chromosome, and that chromosome is going to start to move from the donor cell to the recipient cell. Before this conjugation is done, before it gets the entire chromosome into the recipient cell, your mating bridge is going to break. So here we see a severed mating bridge. This cell will eventually finish replicating and replace the pieces that it's lost. This is not a full chromosome here. It does not come back into a circular chromosome. But what we see are two complementary parts of this chromosome. The original cell, the recipient cell, is shown in green, and the donors is shown in brown. And what happens is these two organisms swap part of their genes. So they swap out those complementary pieces, almost like crossing over in prophase 1. Once that swap has occurred, this organism now has a new set of genes. So it's not all green, it has a piece from the donor cell in it. So it's considered a recombinant. It's not the same as it was before. It has a new combination of DNA. The other thing to note is that once that swap has occurred of DNA, that extra piece that had come from the donor cell will break up and no longer be viable. So it's not going to be in the cell anymore. You would not have an F factor. And so this recombinant bacteria remains a recipient bacteria. It is not a donor bacteria because it does not have the F factor in it. Some bacteria carry R plasmids, and these are genes that code for antibiotic resistance. When you're given antibiotics for an illness, your antibiotics are killing bacteria, but they're also selecting for bacteria with genes that are resistant to the antibiotics because they leave those bacteria behind that are resistant. So those are the ones that survive that condition and reproduce, and they become more plentiful. Antibiotic strands of bacteria are becoming a lot more common, especially when we hear about MRSA in hospitals. So it is definitely a condition that needs more medical attention, but we can use this antibiotic resistance and this plasmid in bacteria to also insert genes and study those genes, which we're going to find through our bacterial transformation lab, where we'll be using a bacteria's plasmid for antibiotic resistance in order to demonstrate that a gene has in fact been transferred or transformed into a cell.